In this video, I'm going to spend some time talking about the neuromuscular junction and a little bit about acetylcholine. I'm doing this because this is probably the best characterized synapse in the entire body. Acetylcholine mediates the message from the nervous system to the muscles here. It's also one of the first it was the first neurotransmitter discovered. So I'm going to spend some time talking about these. These have the advantage of being fairly easily accessible, given that they're outside the brain. You can isolate this synapse pretty easily, and that really helps us study it. The neuromuscular junction is exactly what it sounds like. Here we have a neuron. It's going to be here, coming through, and you can see it branching into several different places. So the terminal boutons are going to branch. When they contact skeletal muscle, this presynaptic neuron will release a chemical we know as acetylcholine. That is going to cause the muscle to contract. Okay? Now, you have to remember this is a muscle cell, not a neuron. This muscle cell acts in many ways like a neuron. Okay, but you've got to remember it's not a neuron in and of itself. So here's a diagram you've seen before, the steps you've seen before. Essentially, our action potential comes down, hits the terminal bouton. Here we get a depolarization. That depolarization causes these voltage-gated calcium channels to open up. Calcium comes into the cell. Calcium is going to interact with vesicles, just like at any chemical synapse. Those are going to fuse and release acetylcholine. So in this case, the red dots are the neurotransmitter acetylcholine. Acetylcholine is going to diffuse across the synaptic cleft, and it's going to interact. Remember, it's going to shake hands with these postsynaptic receptors. In this case, those postsynaptic receptors are nicotinic acetylcholine receptor. Okay, nicotinic acetylcholine receptors. Okay. The reason they're called nicotinic is probably at this point self-evident. Nicotine, the drug in cigarettes, also binds to it. All right. When it binds to it, it gives us allows ions to flow. It turns out this is actually a monovalent cation channel. Okay, so a channel, you know it's a tunnel. Monovalent equals a charge of 1. Cation equals positive charge. I'll also talk about anions. Anions are negatively charged. All right, so a monovalent cation channel is a channel that allows any ion that's +1 to pass through it. Here we're going to be talking about two, sodium and potassium. So at the nicotinic receptor, sodium flows in, down its concentration gradient. Potassium flows out, down its concentration gradient. And that's occurring at the same time. Just like near the peak, just before the peak of the action potential, both sodium and potassium are flowing. Now who wins? It's sodium. So sodium comes rushing into the cell. I get a depolarization. Okay, for this class, all I want you to know, depolarization equals contraction of the muscle. We'll learn a little bit more about the mechanisms in the chapter on movement, but for right now, as long as I have depolarization, I have muscular contraction. Now I've got all this acetylcholine kicking around. I've got all of these nicotinic acetylcholine receptors being activated. So when those are all activated, I have a massive depolarization and my muscle contracts. Okay, that's what causes contraction of the muscle. Sorry, I was drinking water there. Now, We need to terminate that signal somehow. Here at the neuromuscular junction, the way that's done is by an enzyme called acetylcholinesterase. Okay, so there's the structure of the membrane actually looks like these. There's sort of crypts down at the bottom here. So when acetylcholine is floating around here, it'll shake hands, come off, shake hands, come off. When it comes near an acetylcholinesterase molecule, the acetylcholinesterase breaks it down. And that's pronounced ACH, ACE. Metabolizes or breaks down acetylcholine. Okay, so we metabolize the acetylcholine. Now that acetylcholine is no longer acetylcholine, those molecules are just going to diffuse away. Things like astrocytes can pick them up. The cell body can reuse some of those chemicals too. Okay, so now once the acetylcholinesterase has done its job, now we terminate our signal. We lose our depolarization. Without a depolarization, then the muscle relaxes. Okay, so I said this fairly quickly. There's kind of a lot here for you to go through and make sure you understand. Check it carefully. The neuromuscular junction is really important, and we're going to talk about it a lot. This is just another cartoon showing you what it looks like. Here we have our skeletal muscle fibers. We're going to have our postsynaptic membrane here with acetylcholine receptors. Here we've got our crypts with acetylcholinesterase in them. Okay, just another picture showing what it looks like. Now, this is an example of the nicotinic acetylcholine receptor. Normally, the channel is closed. Okay, so blue, these are ions. Blue, there's more of the blue on the outside than there is on the inside. Okay, there's more red on the inside than the outside. So that you should be able to figure out that sodium is blue, potassium is pinkish. And you can look at the key and see, yep, we're right. We do know something. Okay, so these ions want to move down their concentration gradient. Sodium wants to come in. Potassium wants to come out. When acetylcholine binds to the receptor, it changes the shape. It opens that pore. It makes it bigger. So now both sodium and potassium can flow. So you know then that the sodium will try to flow in. Here now, because there's no blockage, we can. Potassium will try to flow out. Again, potassium can flow out down its concentration gradient. Okay, so this monovalent cation channel allows K plus and sodium plus to pass. Sodium wins. And by wins, I mean contributes more. We get a depolarization. Now, one thing I want you to point out, if you look, there's 5 different subunits. It's basically 5 different Lego pieces you've got to put together in order to make the receptor. There's an alpha, beta, gamma, delta. Okay, most nicotinics will have 2 alpha subunits, then beta, gamma, delta. Okay, each of these is one big protein. So when we make nicotinic acetylcholine receptor, we actually have 5 genes for it. Multiple genes for it. Just remember that it's a big protein. In order to make it, we need genes being turned on. Okay, I'll give you one last cartoon about this. The reason I'm sort of going over this over and over and over is, in my experience, students take a little while to learn it. This is so important. Just go and write this out over and over and over. I hope the test motivates you to do that some, but more your ability to understand it relies on this. So we've got synaptic vesicles containing neurotransmitter, right? My action potential comes down, voltage-gated calcium channels open, and calcium comes in. Calcium interacts with a bunch of proteins. That ends up making the neurotransmitter be released. That neurotransmitter is going to go around, shake hands with the receptors. Since we're talking about the neuromuscular junction, you know this is acetylcholine. Okay, and when we have our membrane channel open, right, sodium, potassium going to flow down the concentration gradients, and then we'll end up, as long as the neurotransmitter is bound, we'll have the flow of ions. When neurotransmitter is released, my receptor closes, and I don't get any more flow of ion. It turns out there's two different kinds of receptors. Well, let me rephrase that. There's two major categories of acetylcholine receptors. We have nicotinic acetylcholine receptors and muscarinics. The nicotinics are ionotropic, and those are what's at the neuromuscular junction, what I just went over. The muscarinic is a GPCR, metabotropic receptor. These you find all over the body, but there's tons of them in the CNS. By the way, although I'm talking about it at the neuromuscular junction, there's also a lot of nicotinic acetylcholine receptors in the brain and central nervous system. Okay, so we have two different acetylcholine receptor families. One is going to be ionotropic, the other is metabotropic. They do very different things. When we are talking about sort of rapid effects, we're going to be talking more about ionotropic. When we talk about longer term effects, we'll be talking about muscarinic. Here at the neuromuscular junction, obviously, though, this is the one that is essential. And that's going to be depicted here. Okay, so this is the one we went over. Neurotransmitter, acetylcholine in this case, binds, opens up the pore, sodium flows in, potassium flows out. You know that net activity results in a depolarization here. At the GPCR, this is the muscarinic receptor. Neurotransmitter binds. That doesn't open that. Remember, this isn't an ion channel. It is just a protein that spans the membrane. When the neurotransmitter binds here, it's going to change the shape of this G protein. Right? This G protein is going to start a second messenger rolling. This second messenger then here is going to go and activate other proteins. And we get a cascade of biological proteins. Okay? Now, what happens at this muscarinic receptor depends on where we are and what the G protein is hooked up to. Sometimes the G protein can be hooked up to a potassium channel. So in this particular case, if you look, we have acetylcholine bind that activates the G protein, which comes off here, and it activates the potassium channel. So when potassium channel opens up, potassium flows out. In this case, we get a hyperpolarization. Okay? So you've got to remember what the receptor is, where it is, and what it does. Some of these muscarinic receptors are hooked up to potassium channels. Others are hooked up for other places. Okay, so acetylcholine actually is going to slow the rate of the heartbeat. If we were to drip it right onto the heart slows. You can almost get it to stop. That's because it's acting here in the heart. It's acting with this potassium channel. That results in a hyperpolarization, makes it harder for the heart to reach threshold. Okay, just to show you, when we talk about the metabic conformation, when we talk about the metabotropic receptor, you can see there's no pore, there's no tunnel. I just have a bunch of intracellular proteins hooked up to a transmembrane set of proteins hooked up to some stuff on the extracellular side. When neurotransmitter binds, it's going to change the shape of the inside a little bit, and that is going to activate those G proteins. All right. One thing about metabotropic receptors. Metabotropic receptors amplify signals. So if we start with one receptor has a bunch of G proteins on it. Okay, those G proteins are going to go and activate a second messenger. Now, each of these second messenger enzymes can go and can make a whole bunch of messenger. I can make a whole bunch of cyclic AMP. Those are going to go and activate other proteins. Each one of these can then go on and activate a bunch of other proteins. So the target proteins have lots and lots. You can kind of think about, obviously I was in COVID here, so this is like one infected person can go and infect 20 people. Okay, so we get an amplification of the signal. Don't worry about numbers or anything like that. Okay, but there are steps when we go from the receptor to my target effect. Remember, all the way down here, that's what we really care about, the effect. We see that one receptor has a large effect on many proteins. Okay, now back to the ionotropic receptors. You remember this, I hope. The ionotropic receptors, the ions flow through the receptor it does, like the nicotinic acetylcholine receptor. You can just refresh your memory here too about what each of these do in terms of excitation and inhibition. Now, like I said, neurotransmitters released. Do we want the neurotransmitter hanging out very long? Well, you may think you do. Cocaine is one drug that keeps neurotransmitter from being taken back up and keeps it active longer. People like that. That's fun for people. But really you don't, because if I'm talking about the neuromuscular junction, I release acetylcholine, binds to the nicotinic acetylcholine receptor, sodium flows in, depolarizes that cell, and I get muscle contraction. I don't want that to last all day long. I don't want to stay contracted for a very long time. So you have a way of getting rid of it. And like I said before, that's acetylcholinesterase. More generally, at the chemical synapse, we've got two options. We can take it back up and recycle it. We do that with the catecholamines. So with serotonin, dopamine, norepinephrine, and epinephrine, the way we usually terminate the signal is by grabbing them and bringing them back into the presynaptic terminal. For acetylcholine, though, it relies on metabolism. We have to break it down. Okay, so this is the way it works with, here they're showing dopamine, but this is the same way it works with serotonin, norepinephrine also. Here's supposed to be a synaptic vesicle. Here's my dopamine molecule. Normal action potential has come down. We get calcium. This is docking and then releasing dopamine into the extracellular space. Okay, when it's in the extracellular space, it's going to float around for a bit. It'll interact with the receptor and activate it, come off, float around a little bit more, do the same thing. Now, in the presynaptic membrane here, we have a special protein. What this protein does is it grabs dopamine and then pulls it back into the cell. Now, once it's back in the cell, I can break it down. Things like MAO, we'll learn about that later, or COMT. These can break down these dopamine molecules into component parts. Or sometimes they can get repackaged. We can reuse them. Okay, that's what normally happens. With cocaine, right, we still have our dopamine released. It's interacting with receptors. It works really well with this protein. This protein is called the DAT, the dopamine transporter protein. Okay, so this dopamine transporter protein does exactly what it says. It grabs and transports dopamine. In this case, it does it from the inside to the outside. Okay? So the dopamine transporter protein does. For serotonin, 5-HT, what do you think the name of that protein is that's a transporter? It's a different one. Dopamine transporter is specific for dopamine. Serotonin is specific for serotonin. So how do you think we name this? Yeah. Serotonin transporter protein. It's also sometimes called 5-HT. I mean the HTT, serotonin transporter. Okay, so these two proteins are going to obviously do the same thing. They're going to bring neurotransmitter from outside the cell to the inside of the cell. If we prevent that, what that means is dopamine sticks around longer. Okay, so now instead of a millisecond, it'll stay for, I'm making the numbers up, but it'll stay for 20 milliseconds or something. It'll stay for a much longer period of time. That means I can bind to and activate more receptors. So once cocaine is released, once dopamine is released, the cocaine is going to prevent reuptake. That means dopamine is going to have a longer effect. Back here at the neurotransmitter, or back at the neuromuscular junction, okay, normally my muscle is going to contract, and then acetylcholinesterase will eat up all of the acetylcholine. That'll terminate my signal, and my muscle will relax. So I'm just going to draw this out here for you real quick. So here's acetylcholine floating out. Here we have the postsynaptic receptors. And intermingled, we have ACH ACE. Okay, so ACH ACE plus acetylcholine yields inactive products. Okay, so it breaks it up. So normally, this acetylcholine will shake hands here, move over, maybe shake hands here, then be broken down by acetylcholinesterase. What would happen if we got rid of acetylcholinesterase? Would it affect my action potential? No. Would it affect calcium coming in? No. Would it affect docking? Would this still dock? Yes. Would acetylcholine be released? Yes. What would acetylcholine do? It'll bind to its receptors like normal. When I bind to the receptor at neuromuscular junction, what happens? Muscular contraction. Good. So I have muscular contraction now. Now, normally, acetylcholinesterase would chew up all that acetylcholine really fast, and I'd be able to relax. What if I don't have acetylcholinesterase around? Now, the muscle contracts, and it stays that way. You basically have something called tetany. Tetany. That's a prolonged muscular contraction. Now, if this goes on too long, what happens? Normally, you breathe because you contract muscles to breathe in, and then you relax muscles to breathe out. If I breathe in, and now my muscles stay contracted, what's going to happen to me? Yeah, you're going to suffocate. You're going to die. You can't breathe, basically. Okay, so that's what would happen if acetylcholinesterase didn't work. The muscles would remain contracted. That's called tetany, and that can lead to death. All right, so those are the basics of the neuromuscular junction. We're going to talk a lot about the neuromuscular junction. You have a case on neuromuscular junction. If you have any questions about it, feel free to ask. Thanks for stopping by, and I'll catch you later.